Okay, so far in this video, it's saying that the skin and the tears and the mucous membranes have kept out the pathogen, but if they can't keep them out. So now the pathogen, the bad guy, is about to enter into the body. Okay, so here we are entering in. Okay, so we talked about that yesterday. In most cases, the macrophages, which is said on the video, patrol the body, simply eat the invader. So that was all yesterday's lecture. If it gets past the skin, past the mucus, past the tears, then the macrophages simply eat the invader, right? So did y'all see that hand reach out? What would that hand be? A pseudopod, right? A pseudopod eating an invader. Okay, good. Keep watching. What if that fails? Okay, so this is really today's lecture. After the macrophages didn't help, the helper T cells. Did y'all say that lady on the radio? She was like the helper T cells. So what do you think that the helper T cell does? Yeah, they send out a signal. They activate the other cell. So helper T cells don't kill anything. They simply activate the other cells. So it looks like this is going to be somebody else. The helper T cell was that lady on the radio. So what did it say the antibodies did? Yeah, the antibodies attached to the pathogen. What's the pathogen? Good, anything that causes a disease. So the antibody attacks to the pathogen, so now the macrophage can eat it. So that's what antibodies do. They attach to the pathogen, so the macrophage is able to identify the pathogen better. Good, but even that should fail, if that should fail. Big guns. So the killer T cell is like your last hope of you getting better without going to the doctor, without getting a medicine. So what was it called, those big guns? Killer T cells. Good. Killer T cells are the big guns. So the memory T cells, did y'all see the word memory? There's memory T cells and memory B cells. What do you think that they do? Yeah, so it said they're filing it away if it comes in again. They remember how to kill that one pathogen. So it's much quicker. If you get if the same pathogen infects you twice, it's going to take longer the first time. The second time, you have those memory cells that are just ready to knock it out. A lot of times, when you get sick the second time, you don't even have any symptoms. So you don't even get sick. So like if you and I came in contact with chickenpox today, that virus is going to enter in our body. Boom, we never even got the first little itch. because our memory T cells and B cells knew how to fight it. We didn't even get sick. Okay, grab a sheet of paper. Math quiz. And go ahead and grab, while you're getting stuff out, go ahead and get your immune system part 2 notes out. Dang. Okay, so let's work this. First of all, it tells you that delta H is equal to 59.6 kilojoules. Oh man, y'all can't. Okay, so because it says 59.6 kilojoules, that should have been 59,600. So that's where, I think that's where most of you messed up right there. Okay, so when you plugged it in, all you had to do, after you converted your temperature, that temperature should have been 300. So when you plugged it all in, let me set it up for you. Should have said 59,600 minus 300 times 210. Okay, and then something else I saw some people mess up on was your order of operation. Make sure, because we're using this 4-function calculator, you do multiplication first, and then write down a number, equals, and then type in 59,600 minus that number. Okay, so your total number should have been negative 3,400. So that was your answer that was your calculate answer. How would you explain whether if it was free energy or not free energy? Good, because delta G is negative. Is that spontaneous or non-spontaneous? Spontaneous. So, okay, everybody who got the justification wrong, write that down somewhere at the top of your notes. If delta G is negative, that's spontaneous. And that means there's free energy available. Okay, so everybody write that down for me. If delta G is negative, that means that it's spontaneous and there's free energy. What that means is it's not held up in the bonds. The energy is not, if it's all positive, that means all the energy is being used in the bonds. There's no energy that's available to do work. So the energy, you have some energy that's spontaneous so that you can have your heart beating. Negative delta G is spontaneous, and that means there's free energy. So positive delta G is what? Non-spontaneous, right? So if you miss that, write it down somewhere. Negative delta G is spontaneous, which means free energy. Positive delta G is non-spontaneous. That means no free energy. Because that's something we haven't talked about in a long time. Okay, so from yesterday, let's just go over a couple things. Because today's lecture, it's not hard, it's just a lot. Because yesterday we did innate, today we're doing acquired. Okay, so let's just go over these a little bit. What does innate immunity mean? You're born with it. Somebody, come on, something else. All animals have it. What else?
It's the first and second.
Okay, first and second defense is your barriers. Mucous membranes. Come on, I need better than this. Sweat is an example. I like that.
Sweat and mucus.
Skin, tears. Those are all examples. What was the cell that was the example? The inside, innate immunity. Inside your body, what was that second defense? Macrophages. You got to remember that because today we're going to talk about some more cells on the inside. Phagocytes. And, or remember, you could call it phagocytes. They could be called monocytes. They could be called macrophages. Those three words mean the same thing. Monocytes, macrophages, or phagocytes. Okay. But I still haven't heard one thing about innate immunity I want to hear.
Do you need to know like which one's like blood and which one's tissue or things? Okay.
Come on, one more thing. Is it intentional?
No.
Is it specific?
No, it's not specific.
Good, that's what I want to know. Innate immunity, you're born with it and it's not specific. Hey, your skin is not selective of which bacteria get to enter and which bacteria don't. It's trying to keep all bacteria out, right?
So it's not. Is it permeable or something selectively?
Right, yeah, your skin is not selectively permeable. It is keeping everything out, trying to. Okay, somebody look at your notes from yesterday. What was a priogen?
Fire protein.
Good, causes a fever, right? What releases it? What does WBC stand for? White blood cells. White blood cells release it. Better than white blood cells, macrophages release it. Yesterday on your synthesis question, a lot of people wrote as the innate immunity white blood cells. Remember, I told you to be as specific as you can. Yes, macrophages are a white blood cell, but there's lots of white blood cells, right? Okay, so what was interferon? We watched that video on interferon.
It's like an antibody.
Not an antibody. It's a chemical release from what? Who releases interferons?
It attracts.
Yes. Okay, so 6 cells, infected cells would be a better way to say that. Infected cells release interferon when they've been infected with a virus. Okay? So when a virus infects your cell, it releases interferons, and what does the interferon do? It notifies the surrounding cells. Okay, let's get that because it doesn't let y'all get that. Okay, when a cell is infected with a virus, it releases interferons. When a cell is infected with a virus, it releases interferons, and the interferon notifies surrounding cells to watch out. It's not like a suicide. What was that suicide one?
Isn't that T cells?
That was the first.
Oh, was it MHC1 or MHC2? Oh, y'all don't look sure. MHC1, right? MHC1 is by your cells, like tissue cells, muscle cells, whatever. Your cells that say, okay, I'm infected, eat me. Interferons are when you're infected with a virus, you're just notifying the other cells. Hey, watch out, there's a virus around. Okay, the difference in MHC1 and yes, ma'am.
So are interferons the things that are sent out to people, or
Okay, when I'm infected with a virus, I'm a cell and I get infected with a virus, I release interferon. And remember the interferon, do you remember the video? It bound to the receptor proteins on the neighboring cells, and it blocked the other virus from getting in. So the virus can infect the other cells because the interferon is in the receptor site. Remember I told you that my dad had hepatitis, and he took interferon? But it didn't. And the doctors told us that while my dad was taking interferon, the virus would not infect other cells. Like he wouldn't get any sicker while he was taking the interferon, but it wasn't going to cure him. So the interferon, all it does is help prevent the virus from spreading. But now that's what the doctor told us. But now that you know the cellular level, do you see why? Why couldn't the virus spread while my dad was taking interferon? Yeah, because it was blocking the binding site for the other cells, right? But the interferon made him nauseous, so he couldn't take it a lot.
They not have anything that can kill them. So
Not hepatitis C. Mm-mm. Nope. Okay, MHC1 and MHC2. Somebody tell me the difference. Good. Perfect. She said it perfect. MHC1 is when, like, tissues, muscles, anything but white blood cells hold up a thing that say, I'm sick. Kill me. Somebody needs to eat me because I'm infected. This is like your surrender flag. MHC2 is shown by white blood cells. Better yet, shown by macrophages. MHC2 is shown by macrophages, and it says, look, this is what I killed, now you go kill it. You got it? Okay, inflammation. Somebody tell me the process. What causes inflammation? Yeah, the blood is pooled in the infected area, and there's a fever. Why does the fever come? What does the fever do? It speeds up the macrophages, right? And the virus can't live in that temperature. So 2 reasons why it gets hot in an infected area. Number 1, it's trying to kill the bacteria with the temperature. Number 2, when it's hot, the macrophages go faster. Okay, and then what did you remember about plant immunity? Thorns. Okay, everybody said thorns. Come on, something else. Thorns, cork, cuticle. Some people on their synthesis question wrote about the plant cell wall. I thought that was a pretty good answer. Plant cell walls probably keep out some pathogens. Poison. Some plants have a poison that they release. Good. Okay, so today is all about, so look at your notes for part 2. Today is all about acquired immunity. Things you should already know is signal transduction. What are the 3 steps? Reception, transduction, response. Y'all got that? Good. Okay, so today we're going to talk a lot about signal transduction, how white blood cells communicate with each other. And then it says glycoproteins and glycolipids are part of the ECM. What is the structure of a glycoprotein and a glycolipid? Good. A sugar is attached to a protein, right? A sugar attached to a protein. Or a sugar attached to a lipid. And what do?
They feel.
They feel, right? They feel, but better than feel. They identify. I like that word better. They help to identify or recognize. They are the feelers. Okay, looking at the first part, it says antigens and immune response. I told you yesterday, what is an antigen?
A surface protein on a pathogen.
Good, a surface protein on a pathogen. Come on, you remember that. I thought I had a picture of it, but I guess I don't. An antigen is the normal protein on the outside of a cell. It's the normal protein on the outside of a cell, but your glycoproteins can recognize it. That's what they feel. Yesterday when I was trying to get my last period class to guess what I was doing, I was trying to get them to say the word identify, just like I do y'all. Somebody said, it's like they're reading Braille. And I was like, that's exactly it. The glycoproteins are reading the what? What is the Braille? What am I feeling on her? Well, yeah, I'm feeling her hair, but what am I really... if she was a pathogen, what am I feeling? The cell wall, the cell membrane, and she has proteins on her cell wall that help me to identify her. And I'm like, ooh, this is a bad one, right? Because my glycoproteins allow me to feel her antigens. And I'm like, oh yeah, that's definitely a bacteria, not just a regular red blood cell. Because your white blood cells, your macrophages need to know the difference in, okay, this is a bacteria, but this is just a normal red blood cell. He can stay. You see what I'm saying? Okay, so antigen is a surface protein. Antigen receptors. Okay, all the antigen receptors are the glycoproteins and glycolipids. Antigen receptors, those are the feelers. They're the identifiers. You might want to write in the word identify there. They're the feelers. Antigen receptors recognize when a pathogen, okay, so next line. When a pathogen is identified, so okay, I'm feeling right here with my glycoprotein, I recognize that this right here is something bad. This surface protein, this antigen is bad. Then it says when a pathogen is identified, it triggers the immune response. It says the clonal selection, but it triggers your immune system to start working. The clonal selection makes those memory cells. So it's going to start making cells. Okay, and here you can see this yellow part right here, this yellow cell. That is like a pathogen. And those pink things on the outsides, those are their antigens, their surface proteins. The green things we're going to talk about in a second, those are antibodies. Okay, so primary immune response and secondary immune response. We already talked about this. The first time you get sick, the first time you get a certain virus, it may take 10 to 17 days. So your first encounter, 10 to 17 days. Your second encounter, only 2 to 7 days. So Here's a picture showing you that there's different antigens on this bacteria. Oh. See this bacteria? So that brown thing is the bacteria. There's different antigens on it. And then it says the word epitope. The epitopes are the tips of the antigens. So basically how the antigen feels is called an epitope. What does that word mean to you, epitope?
Above.
Yeah, on the very top. An epitope is what the glycoprotein or glycolipid feels. Did I mess up your hair? Sorry.
No, my headband.
Okay, do me a favor. Out beside where it says primary immune response and secondary immune response, just make a graph, see what you think. So draw just a little graph expressing the primary immune response and the secondary immune response and justify your reasoning.
Hey, do you ever watch Prime?
Mm-hmm. Yeah, this was on the video last night if you watched it.
Because I need it.
But I want two different lines, like the first time and the second time. Or you can make it one long line if you wanted. That's up to you. Yeah. Your body already knew how to make antibody. What is an antibody? Fights off an infection. Okay, so I told you I was going to draw it another way too. Another way to draw this would have been if you had one line. I think that's how it was last night on the video. So if you had days down here, and response here, the first exposure would look like this, and the second exposure would look like that. So that would be if you had one line. Still shows the same thing. Okay, so on acquired immunity, you have lots of parts to acquired immunity. The first thing you have, if you look at your notes, it says the humoral. What did it say last night on the video about humoral? In the fluids, right? So where it says humoral, you have, that's your immunity that's floating around in your lymph nodes or in your blood. And then you have cell-mediated, which is on the other side of that graph. And cell-mediated is cell to cells. I'm going to kill you. Your cells kill your cells. Why? Why would your cells kill your cells? Because they're infected. So that's going to be where that MHC1 comes in. It kills because of that. So, but you have two types of lymphocytes, two types of acquired immunity, white blood cell that we're going to talk about today. We're going to talk about three. You have T cells and B cells. Okay, this is easy. B cells make antibodies. B. See the B in antibody? B cells make antibodies, and they're made in the bone marrow. B, and B. B cells make antibodies. That's one word. That's got to be in it. And it's made in the bone marrow. Okay, T cells are made in the thymus, Thymus, and they release a toxin. T cells are made in the thymus. The thymus is right next to your heart. Thymus gland. T cells make toxins, and they're made in the thymus. So they're really made in the bone marrow, but they're formed in the thymus. Like they're made in the bone marrow, then they go to the thymus, and that's where they grow up to be adults. So then they're released from the thymus. So T cell, thymus, toxin. B cell, antibody, bone marrow. You got it? And those are all inside your body. Okay, looking at the first one, it says B cell, you want to highlight antibody, and you need to write in made in bone marrow. T cell, you want to highlight, chemicals, and then write in formed in the thymus. Okay, there's two types of T cells. There's helper T cells. What did you know from the video? What do they do? T cells, what do? They call for help. Help, help, help. Remember the little girl on the radio? Help, we're sick. We got an invader. Helper T cells don't kill anything. They call for help. Just like that girl on the little radio. She didn't kill anybody. She just called to notify the other ones, and it says cytotoxic T cells. Okay, do me a favor. Write in killer T cell. Because sometimes they're called killer T cells. Cytotoxic T cells are killer T cells. They're the ones that really bring out the big guns. They release a toxin that kills. Okay. The B cells and the antibodies are floating around in the fluid. B cells and antibodies are floating around in the fluid, and they just come into the pathogens. Okay, T cells feel. They're cell to cell. So that's what we call cell mediated. I'm sorry, I keep touching. Right? So B cells are in the humoral. T cells are the cell mediated response. Okay, so look on down where it says humoral, highlight fluids, highlight antibodies, highlight B cells. Okay, so look at this. This is like the coolest picture ever. So you can see this. The yellow thing is the pathogen. And then I guess those little red things are the antigens. Those are the surface proteins. But do you see how these little turquoise things, they're shaped like that? That one's upside down, the one I circled. But can y'all see how they're shaped? That's what an antibody looks like. And what the antibody does is it attaches to the antigen. So it attaches right there. And really, the pathogen can't work when it's got all those Y-shaped proteins on it. So antigens are the surface proteins that antibodies attach to. So an antibody looks kind of like a tongue right here. And what it's going to do, if Jessica right here was a pathogen, how well could she work as a pathogen if this was on her head? But wait, what if it was all over her? She wouldn't be able to work very good, would she? Okay, now, if you were, sorry Jessica, if you were a macrophage and you were feeling and you felt a whole bunch of these, would you not be like, oh gosh, that's something bad? Right? So antibodies are produced and they attach to the pathogens like this. And then the macrophages, or the killer T cells, can feel better. So really all it is a marker. It's an identifier. Hey, right here, this is a sick one. Going around and their antibodies just mark them. Okay, so looking at your notes, you want to highlight humoral, we said fluids, antibodies, B cells. B cells, it says become plasma cells. The plasma cells is a memory cell. The word plasma cell is a memory cell. Okay, B cells are activated by, you've never seen interleukin 2s. B cells are activated by interleukin 2s. Okay, look at that word, interleukin. What does that word mean to you? Inside. Okay, pull out your list. It's L-E-U-K on there. See if it is. I don't know if it is or not, but it should be if it's not. Yeah, it is. White, right? So if inter means inside or within, and leuc means white, now let's guess what interleukin 1 means. Between white blood cells, right? So we're talking about the immune system, so it's white blood cells. Interleukins is communication from white blood cell to white blood cell. In other words, it's a ligand. White blood cell to white blood cell. That's the language they speak. They speak interleukin, ligand. So interleukin, right in there. Interleukin 1 is the first communication between white blood cells. What do you think interleukin 2 is? The second communication between white blood cells. So interleukin 1 is the first one. Interleukin 2 is the second one. Okay, so go back up to where it says humoral, under humoral immunity. On A, it says interleukin 2 is released from T helper cells. Think about the video. You know that girl you saw on the radio? When she was speaking, she wasn't speaking English. She was speaking interleukin 2. So helper T cell releases interleukin 2. You with me? Helper T cell in the radio released interleukin 2 to notify B cells and to notify T cells. What does interleukin 2 say? What'd that girl say on the radio? There's an invader. Hey, I need some help. So interleukin 2 says, there's an invader, I need some help. So helper T cell releases interleukin 2 to activate T cells and B cells. So that's how you would say that in a paper if you're writing it. Helper T cell releases interleukin 2 to activate B cells to do what? Make antibodies. And T cells to bring out the big guns. We haven't talked about T cells yet, but that's what they do. Okay, so go on down to cell mediated. That's where cells touch cells and they talk about it. Cells touch cells. So it says cell mediated, that's going to be your T cells. Highlight the word T cells. You got two types of T cells. Killer T cells and helper T cells. Helper T cells do what? Call. They call for help. They don't kill anything. They're the little skinny girl on the radio. They can't kill anything. They just release interleukin 2, right? Okay. Cytotoxic T cell, that's like your last line of defense before you have to go to the doctor. You're not going to get better. If your killer T cells can't handle it, you need to go to the doctor. Okay, so it says, highlight, cytotoxic T cells mature to fight and kill infected cells. T helper cells initiate 2 types of specific immunity. Okay, so T helper cells, they release that interleukin 2 to call on them, but they respond to interleukin 1, which is released from macrophages. And I'm going to draw this out, and I think it'll be easier for you once you see it drawn out. Cytotoxic T cells, okay, so the killer T cells, the big guns. What do? And we wrote in killer T cells. Cytotoxic T cells and killer T cells are the same thing. Okay, so they, on B, release a toxin called Perforin, P-E-R-F-O-R-I-N. And it makes holes. Y'all see that word? It's like, with their toxin, they drill holes in your infected cells. So where antibodies attach to the pathogen, Jessica was a pathogen when I put that tong on her head. She was a pathogen. Okay, killer T cells kill your cells that are infected. So when they hold up their MH1, killer T cells blow holes in them. Yep. Now you're like, oh, that's why I'm tired when I'm sick. Because you're spending all of your energy with these cells talking. You know how when you get a cold, you're just tired? Just like, I just want to lay down. That's why. Because you got all these cells making memory cells and trying to make antibodies and release these toxins. You're spending all your energy doing that. You can't spend your energy playing basketball. Okay. So on cytotoxic T cells, do you see where it says they are activated by MHC class 1? What does that mean? What's MHC class 1? I'm infected, kill me. And what is IL-2, interleukin 2? Where did that come from? That's from the T helper cells, right? Okay, look at this picture real quick. So this is showing you, right here, this is your infected cell. That's your cell infected. It looks like it's got those little green and blue things inside it, which is infected. Okay, so your cell holds out the MH1. Y'all see the MH1? That's your cell saying, I'm sick, I'm infected, come on, just kill me. Okay, the T cell right here, cytotoxic T cell, feels it with its, this is a glycoprotein it fills it with. I'm about to tell you one more word though. The helper T cells and the cytotoxic T cells, their glycoproteins are called CD4s. That's just a little by the way. If you ever see that, that's what that is. So they're feeling with their glycoprotein. So right here, that's the T helper cells glycoprotein. Okay, and then this one shows you right here. This is the helper cell. So these are both T cells. This is the T helper cell with its little CD4. And it also feels, this is the macrophage. And it's going to engulf the microbe. And do what with it when it engulfs it? What's it going to do once this purple thing is a macrophage? What's it going to do once it engulfs the microbe? It's going to break it up in its lysosome, right? Okay, and then it's going to present its MHC2. So this picture is showing you the difference in 1 and 2. This is your cell that's sick. See, it's sick. Oh, it's sad. And this one is the macrophage. This one's a white blood cell. You got it? Sort of? Okay, so an antibody looks like this. Antibodies have, this is the last part of the notes. This is the antibody right here. Ooh. Right here, attached to the antigen. What's an antigen? Come on, what's an antigen? Surface protein, right? Antigen is the surface protein. Antibodies attach. So this is Jessica right here when I attach that to her. Hey, Jessica. Jessica was an infection. She was a bacteria, a pathogen. And antibodies attached all over her. How hard would it be for this bacteria to work? Sorry, Jessica. I'll take your name off of it. How hard would it be to work if you had antibodies connected all over you? Somebody tell me, what is an antibody? Make me proud. What is an antibody? It's what? It doesn't fight. It's a marker. An antibody is a marker, right? But what is it? What's its structure? A protein. And yes, a Y. You're right. It's the structure of a Y, but it's a protein. What are proteins made of? So, okay, let's say, what is the structure of an antibody? You tell me. A Y-shaped chain of amino acids, right? It's folded. Its quaternary structure is a folds of amino acids. So it says, one thing you want to know, the word antibody also means immunoglobulin. Same thing. Immunoglobulin, that's what doctors call it. We call it antibodies. Same thing. They are, highlight the word protein. Okay, so antibodies, look at this picture. And I think you looked at this in your drawing last night. They have a heavy chain. Right here. Heavy chain. These are linked by, where have you seen disulfide bridges? The tertiary structure. Do you see those bridges right there? Disulfide. 2 sulfurs attached. It's a bond with 2 sulfurs. Disulfide bridges using cysteine, the amino acid. What do you know about amino acids? They make up proteins. How many of them are there? 20. There's 20 of them. So cysteine is just one of them that makes the disulfide bridges. So cysteine has sulfur in it, obviously. Cysteine, remember the R group? You should know all about amino acids. They have that R group on the bottom. Okay, well, cysteine is one of the 20 things that can attach. Okay, so looking at my picture, it says heavy chains and light chains. They're attached by disulfide bridges right there. Then it says this is an example of highlight, tertiary and quaternary structure. What does that mean? What's tertiary and quaternary? Folding, right? The folding. Yeah, tertiary is where they just crumple up, and then quaternary is where they group together. And then it says there's a variable region and a constant region. What that means right here? Nope. Maybe if I go to the end. Nope. Okay, right here, this is, look at my picture. This is all constant. Okay, but this right here varies. It may be shaped like that. Or it may be shaped like that. Why does it vary? Why is it shaped differently? Yeah, so the antigen that's shaped like this can fit there. And the antigen that's shaped like this can fit there. So the variable region is because of the different antigens. Okay, this is a picture of what your graph should have been like. But this is the one line. So you've got first exposure of antibodies. Second exposure is shorter and you make way more. So it shows you, if you look at the first exposure, it's like from 1 to 28 days. The second exposure is like 28 to 35 and you're better. So 28 to 35, is that 35 maybe? Something like that. Maybe 40. And you're all better. And this is all because of memory cells. Memory cells. Okay, grab your sheet of paper. I'm going to draw this out for you and see if we can make more sense of it. Okay, so this is a drawing of what we just did in class. And you may need to come in for help because this is a lot of information. But what I'll do is I drew it out. And we have here the innate. And we went through that first. And that's what you're born with. And you've got skin and mucus and normal flora. And those are like barriers. So I'm looking at number 1 right there. And then number 2, you got the saliva, tears, the pepsin, that's the chemical in your stomach, and the sweat. And those are a change in pH. So those are all innate immunities. But the third one I drew in a cell. And that's the phagocytes. And they eat the pathogen. So if you ever see monocyte, phagocyte, or macrophage. Same thing. They're all innate immunity, and they eat pathogens. So that's what they do. They eat pathogens, but they present with their MHC2s, the antigen. So they present the antigen with their MHC2s. Okay, well, pathogens have on them antigens, and those are the protein surface markers. Okay, so I'm going to make my ligands, or my chemical messages, in purple. Okay, so macrophages, after it presents, remember, look what I have killed. I have killed this. Now you go kill it too. So this right here, that's to communicate with other white blood cells. But in addition to communicating with other white blood cells, like with other macrophages, this is to communicate with other macrophages. It also releases a chemical message called IL-1. And remember, we said enter between. Luke means white. First message. So the first message is the IL-1. We'll leave it like that. And it's calling for help. Hey, helper T cell, I'm going to need some help. So helper T cell, I need it. Macrophages tell helper T cells with IL-1 that they need help. So now helper T cells are going to release IL-2, which notifies killer T cells, and B cells. Right, so if you look at my picture here, IL-1 is from the macrophage, and it tells the helper T cell to release IL-2. So there's the IL-2 right there. IL-2 is the message from the helper T cell that notifies or activates the killer T cells, so these are killer T cells, and the B cells. So cytotoxic T cells and killer T cells are the same thing. So now they are activated. Okay, so let's talk for just a second about killer T cells. Killer T cells, once they're activated right here, they release paraffin, which is a chemical that drills holes in your infected cells. They release the chemical which drills holes in infected cells. That's why this is cell-mediated. So helper T cells activate killer T cells with IL-2, and that tells IL-2 to release paraffin. All this is reception, transduction, response. If you look right here, you've got reception, IL-2 binds. So reception, IL-2 binds, transduction, and then the response is to create the paraffin. Okay, well right here, same ligand, IL-2, different receptor, reception, transduction. The response is going to be to make antibodies. And you can see my antibody I've drawn there. Well, antibodies don't kill anything. Antibodies attach to pathogens. So if here's a pathogen, there's a bacteria. There's its antigen, its surface protein. Antibodies attach. So there's the antibody. Antibodies attach to the pathogen. And all they do is they mark it. Well, why would they want to be marked? They're marked so that now macrophages or killer T cells can kill it. Okay, so helper T cells, all they do, they don't kill anything. Helper T cells activate B cells, and they activate killer T cells to do the work. B cells mark pathogens with their antibodies. T cells, killer T cells, release the chemical. So what two things kill the pathogen is going to be, first, your macrophages, second, your killer T cells. Okay, so somebody, let's erase all this mess. Somebody with AIDS. AIDS is a virus. If I can get all this erased. AIDS is a virus that infects your helper T cells. So someone with AIDS has an infected helper T cell. So that means that all this still works, but none of this is any good. So without helper T cells, look, without helper T cells, then killer T cells would never be activated, and B cells would never be activated. Macrophages can't do it alone. So someone with AIDS or with HIV doesn't have this helper T cell. So all they have working for them is the innate immunity. So if someone with AIDS doesn't die from HIV, they die from pneumonia or strep throat or something that normally we would be fine with because we have all of the acquired immunity. And someone with AIDS, they have an acquired immunity. It just doesn't work because their helper T cells are infected. So, like I said, this is kind of difficult. This is a lot of information. If you need to come in for extra help, do so. Here is your synthesis question for today. So pause the video, try to write that. And here is your answer. So 3 points possible.